Thank you so much for coming out this morning, everyone. My name's Alex Sooth, but I'm going to be talking about work that was done by my colleague, Leila Wehbe. So let me jump into it. So in a typical controlled fMRI experiment, this is sort of the classical SPM way of doing fMRI experiments. We might see things like this, where we have one condition that is abstract words, another condition that's concrete words. We compute a contrast between these two, and we get some kind of map, right? We find the places that are significant, and we declare victory. Or this might be animate words and animate and non-animate words, or auditory words and visual words. And in all of these cases, we're generating these sort of single maps, but each of these requires a separate experiment. So the questions that we're sort of asking today are we using neuroimaging efficiently when we do these kinds of experiments? That's a leading question, maybe. And do the results of controlled experiments generalize to real life or natural language use? So I'm coming at this from the perspective of natural stimulus experiments. So let me describe to you one of the experiments that we do. We have people listen to hours of naturally spoken stories. So they're listening to actually stories from the Moth Radio Hour. And these are natural, they're uncontrolled. It's just people listening to speech for hours. And then we build computational models that predict the brain activity in each voxel as a function of a sort of high-dimensional representation of the meaning of the words in these stories. And And then the result is that we have, we can predict activity in a large part of the brain using these models. So on the top here, this isn't working. So the brain on the top is showing prediction performance of this model on a held out story that wasn't used to train it. And on the bottom, it's a visualization of sort of which types of words cause activity in which areas. So this is our sort of naturalistic version of these controlled experiments. But now we want to ask, can we combine these in some way, right? So do the naturalistic experiments, do they make more efficient use of information in these controlled experiments? Do they actually recover the same information? And maybe we can build a bridge between these two different worlds. Okay, so introducing, this is the main point here, bold predictions. We want to make bold predictions. That's a pun. So it's automated fMRI simulation engine that's based on these naturalistic fMRI experiments, but where we can simulate controlled experiments. So this is our website. You can go to it now, and you can run your own experiments. You can simulate your own experiments here. Let me tell you a little bit about how it works. So you can enter two sets of words on this website, for instance, concrete words and abstract words. Then these words get projected into the semantic feature space that we use to fit the models for the natural data. The feature space gets projected onto the regression weights. These are actually the models that we fit. And from that, we get a predicted activation map for each of the two conditions. We can subtract those two maps, and this gives us a predicted contrast. So this is essentially what our natural stimulus model predicts the contrast would look like if you had actually done this experiment, right, of concrete versus abstract words. So we can use this for exploratory analyses, like this one, where this isn't an actual experiment that I'm not sure, I don't think people have actually done this before. So we can contrast, say, place words. So this is words that describe places like neighborhood, street, house, building, etc., versus just baseline. And this is showing the predictions from one subject. And one of the really sort of salient things here is that the three classical sort of visual place areas, the retrosplenial complex, the TOS or occipital place area, and the parahippocampal place area come out as very strongly preferring place words to baseline. If we look at an average across, this is just a volumetric MNI, average across 8 subjects predictions, we see a similar picture. So this seems like a sensible sort of result. So this is encouraging for this kind of simulation. And we can also use this tool to do simulated replication, right? So we can replicate in silico experiments that have already been published, as long as we have the stimuli for them. So what we do is we take the stimuli from these experiments, we put them into our simulation engine and use that to simulate the contrast, and then we can compare it to the reported ROIs from the study, which is a tricky detail that I'll talk more about in a moment. So I'm going to give 3 brief examples of this. One is this paper from Jeff Binder from 2005 that compares concrete and abstract word representations. So here's our simulated result for this contrast. So the brain map here, this is showing a flattened representation of a FS average brain with an average of 8 subjects projected on it. The gray voxels, the voxels showing the curvature map, are they weren't significantly predicted by our models, so we don't think that we can say anything reasonable about them. And the colored voxels are colored according to the simulated contrast. So they're red, where we expect the concrete words to activate that area more than the abstract words, and blue for the opposite. And we took the reported activation peaks from the paper and turned those into small volumetric ROIs and then projected them on the surface also, and that's shown in the green hatching here. So you can see that we get some match, especially in sort of posterior parietal cortex and a little bit in prefrontal cortex, but there are also some mismatches, but that's how this goes. This is a little tricky to actually evaluate. If we look at the opposite contrast, so this is the abstract versus concrete, so this is the places that are more activated for abstract and concrete, this looks very different between our results and the actual result. But we think we actually have a very good reason for that, which is that in our study there was no task. The subjects were just listening passively to these stories, whereas in this study there was a task where people had to make responses for each word, and there was a huge task effect actually between abstract and concrete. So if we look at this, we actually just created a map of what the sort of task effects should be using neurosynth data, and it matches quite well where this patch of activation in that study is. And they also show this in a figure in the paper, that the reaction time difference between the abstract and concrete categories really well matches this patch. So we don't feel too bad about missing on that one. Let me show you a second example. This is a little bit different. So this is not a study that was directly intended to look at sort of semantic differences between classes of words, but this is a study where the authors were examining how challenges to political beliefs or statements that challenge political beliefs, do they cause different brain activation than statements that challenge factual or like non-political beliefs. And we just took the stimuli from this experiment, or the ones that they provided, and plugged them into our engine, and we actually get, I think, a pretty decent match to the reported results. So again, the reported ROIs are in green, and the red voxels here are the ones that we expect would have a high contrast. Yeah, so that works decently well. There are definitely cases where it does not work so well. So this is a third example, a study looking at representation of event nouns as compared to object nouns or verbs. So let me show you one example here. So this contrast from the study is event nouns versus object nouns, where event nouns are things like wedding or accident or things like this. They're nouns that describe sort of happenings. And they found a large activation in the sort of posterior temporal cortex, and we just didn't see that in our data. So this was a less good result. So were these results actually replicated? They look good in some cases, but much less good in others. And we're still trying to figure out exactly why that is. You know, do they not replicate because we're using naturalistic stimuli, these narrative stimuli? Or are there just differences in these analysis methods? I think another real difficulty for us is actually we would love to compare to the real contrast maps from these studies, but those are just not available for most things, right? Most studies don't have published contrast maps on things like Neurovault, which would be fantastic if they were there, so please put your things on Neurovault. But we have to try to recreate them from the peak activation coordinates, which is a sort of losing proposition. So let me go back to the 2 questions that I started with. Are we using neuroimaging efficiently? And I think I'm sort of arguing here that naturalistic experiments are more efficient because many contrasts can be computed from one experiment, right? So I showed many examples today, and you can try on your own many others that are all, they all come from this one experiment that we did. We didn't have to run many experiments to get all these different contrasts. We're just doing one experiment. And the second question was, do the results of controlled experiments generalize to real-life language use? And that is more tricky to answer, and I don't think we have a good answer for that one yet. We can say that the results are pretty encouraging, but it's difficult so far. Like I said, we don't have access to the entire contrast map. Another big issue for us has actually been getting access to the stimuli for many experiments. They're not commonly publicly available, and that would be very helpful for us. So I would encourage you, I don't know, send us your stimuli if you are publishing a new, I don't know, that's a strong ask, but if you can put your stimuli into this website, Bold Predictions, you can see what our prediction for your contrast should be. So just let me conclude. We think that naturalistic stimuli can recover more information, so they're more efficient per sort of experimental time used. Does this mean that we don't need controlled experiments? I think absolutely not. But I think there's a less good case to be made for using controlled experiments for exploration. So for sort of trying a new contrast just to see what's there, that doesn't seem like the most efficient use of your time. So what we suggest is that you should use that for sort of confirmatory. We should explore using these naturalistic data sets, and then once we find something that we think is maybe interesting, we can go back and try to confirm it using a real hypothesis test or controlled experiment. So thank you all for listening. You should, again, check out this website, boldpredictions.gallantlab.org. And this work was, again, largely done by Leila Wehbe, who unfortunately couldn't be here today. Please email her if you have questions. Also, come see our poster. I think it's number 1838. I'm going to be there this evening, standing there. And I wanted to mention briefly that all the visualizations on the website and in the talk today are done using this visualization package that we created called PyCortex, so you should check that out too. Thank you very much.
Are there any questions?
Thanks for the talk. First of all, I think the asking for making the stimuli available and the data available, I don't think that should be a big ask. That should be the norm. If we want to make our science reproducible and reusable, efficient, that's what should be the norm of the community. But I had a quick question on how do you test for whether the study has actually replicated or not? You seem to be just visually looking at the differences and have a eyeball thing. Is there any statistical technique that you could use for trying to see whether there is an actual replication?
Yeah, thanks, JB. That's a great question. So we can do statistical tests on this, certainly. So we can actually, I think, interestingly, Leila came up with a really cool way to do statistical tests within this tool that is a much better test than you could normally do in a normal experiment, which it's a permutation test where you actually scramble the stimuli across the conditions and then ask how big is this difference between our actual partitioning of the stimuli versus a random partitioning, and so we can assign significance that way to each voxel's activation, and then we can look at true positive rate and false positive rate in terms of overlaps of these ROIs, but we've shied away from doing a lot of that so far because I think just our reconstructions of the contrast maps based on the sort of peak activations, they just don't look good. Like they don't actually look anything like the contrast maps in the papers, so that's why we haven't really gone deeper on that so far, but that's a great question. Thank you.
Thanks again for the very interesting work, and thank our speaker one more time.